Welcome to Unit 6 of Grade 10. The topic is Structure and Functions of the Plant and Animal Cell. Here, I will do the lesson in the form of answering the questions which appear on your screen. So remember, I have prepared all these questions directly according to the syllabus. So the first question, part A, who was the first scientist to observe a section of cork using a microscope prepared by him and to name the small units as cells? Actually, the name of the scientist is Robert Hooke. So he was able to observe a section of cork using a microscope prepared by himself, and he observed these cells like the chambers in a beehive, and he was the first person who named these chamber-like structures as cells. Then, depending on his findings, another set of scientists continued his experiments and they were able to present the cell theory. So the names of these scientists are Schleiden, then the next scientist is Schwann, and the third person is Radolf Vacuole. The contents in the cell theory are very important. You have to remember those very well. First, they told the cell is the structural and functional unit of life. The cell is the structural and functional unit of life. Second one is all organisms are made up of one or more cells. All organisms are made up of one or more cells. Third factor is new cells are formed from pre-existing cells. New cells are formed from pre-existing cells. If I explain these three contents, when we say the structural unit, for example, we'll take a building made out of building blocks. There the basic structural unit is a single building block. Likewise, in the cell theory they say that the structural unit of life is the cell. All the living organisms, all the living bodies are made out of cells. That is the basic unit. And functional unit of life is also the cell. That means the cells in our body or the cells present in all the living organisms have different functions. If we take examples from human body, the neurons or the nerve cells are responsible for the transmission of impulses. And if we consider the red blood cells present in the blood tissue, they are responsible for the transportation of oxygen to the cells. And if we consider a muscle fiber or a muscle cell, they are responsible for the movements in the body. Therefore, each and every cell type is responsible for a particular function. Therefore, we can say the cell is the basic functional unit of life as well. Then, all organisms are made up of one or more cells. The organisms which are made of one cell, we call them as unicellular organisms. And the organisms like us who are made of more cells, in our body there are several millions of cells. So we are known as multicellular organisms. Then, a new cell is always formed from a cell which was existing earlier. So at the end of this lesson, we are going to discuss about the cell division process, where two or more cells are produced from a from an existing cell. Right. Then we'll move on to the next question. Define the term cell. So according to the things that you have learnt up to now, you can say the cell is the basic structural and functional unit of life. The cell is the basic structural and functional unit of life. Then you are asked, who are unicellular organisms? Give examples. Unicellular organisms are the organisms which consist of a single cell. As examples, we can say all bacteria are unicellular. You can take certain examples like Lactobacillus, then Acetobacter, then there is Vibrio cholera, all these are bacteria, and as these are scientific names, we have to make them italic, the names. And there are protozoans like amoeba. Then there is paramecium, which has the shape of the sole of a shoe. Then there is euglena. All these are protozoans, and in addition to that, we can say there is Plasmodium, who is responsible for causing malaria, and algal varieties like Chlamydomonas, so here you can revise the knowledge that you have gained in grade 9, the first unit. So all bacteria, protozoans and certain algal varieties are taken as examples for unicellular organisms. Then part C, you are asked what is a typical cell. Now we are going to learn about different organelles present in a cell. So for the convenience of study, we consider a cell which contains all these organelles, though actually all the organelles are not present in all cells. So a typical cell is a cell which contains all the cellular organelles. Then in the laboratory you will have to observe examples for animal cells and plant cells. So we are considering cheek cells as an example for animal cells, as it is easy to obtain. So writing the steps to be followed in order to observe cheek cells is very important. You have to remember these steps because these have become popular questions in past term test papers as well. First you have to wash your mouth, and you can take a clean yogurt spoon or you can take a toothpick. Then you have to scrape the inner side of the cheek using the yogurt spoon or the toothpick. Then you have to obtain a clean glass slide and put a drop of water on the glass slide. Instead of water, you can use glycerin as well. The purpose of using water or glycerin is to avoid the cells being drying off. You have to transfer the specimen, that means the cheek cell sample that you have taken, onto the slide on which there is a drop of water. Then you have to cover the specimen using a cover slip. There you have to be careful not to allow any air bubbles. Then you can observe this under the compound light microscope. So to get clear observations, before you cover this with the cover slip, you can use a stain like methylene blue solution. There, after putting the sample on the drop of water or glycerin, you have to add a drop of methylene blue solution on it. Then you have to cover this with the cover slip, and by using a blotting paper, you have to remove the excess liquid. So if I repeat the steps, first you have to wash your mouth and scrape the inner side of the cheek using a clean yogurt spoon. Then obtain a clean glass slide and put a drop of water on it. Then you can add a stain like methylene blue solution. Transfer the specimen onto the slide. Cover the specimen using a cover slip without allowing any air bubbles. Remove the excess liquid by using a blotting paper. Then observe through the light microscope. So here you are shown the steps. Adding water or glycerine is shown here, and the stain is also added in the same manner. And when you are covering it with a cover slip, you can make use of a needle like this. Then here you are shown how to obtain the cheek cells and how you keep the sample on the drop of water or glycerine. And this is the appearance of the cheek cells. Then we'll move on to the next question. Accordingly, you are asked to label the parts of animal cells. So I have given an already labeled diagram. You can see the outermost cover of these cells is the cell membrane, and this dark spot inside is the nucleus, and inner to the cell membrane there is the cytoplasm. So if you are drawing a specimen, as you see under the microscope, you have to state the magnification. First you have to mention the magnification of the eyepiece, then the magnification of the objective lens. Then after seeing it under the microscope, you by yourself magnify the image to a certain extent. Then you have to mention that value also. Labeling the diagram is also important. Then we'll move on to the fifth question. Cells of an onion peel can be observed to study about plant cells. Write down the steps to be followed here. So here I have shown the steps. First you take a fleshy part of an onion. Then by using a faucet, you can obtain the transparent thin section just inner to this fleshy part. Then you can transfer that thin transparent section onto a watch glass containing water, and then if you wish to stain the specimen, you can use a stain like safranine. So what you have to do is transfer the specimen into a watch glass containing a drop of safranine with the help of a paintbrush. Then after keeping it for a while, again you have to transfer the specimen into a watch glass containing water, that is to remove excess stain. After that, you have to take a clean glass slide, keep a drop of water or glycerin on it, then transfer the specimen onto that drop of water or glycerin, then cover it with a cover slip with the help of a needle without allowing any air bubbles. After that, you have to remove the excess liquid by using a blotting paper. Then observe it under the low power of the microscope. So if I repeat the steps for you to write, obtain a thin transparent section from an onion, transfer it into water in a watch glass, then transfer the specimen onto a drop of safranin in a watch glass, again put it into water. After that, obtain the specimen with the help of a paintbrush onto the drop of water on the glass slide. Then cover the specimen with a cover slip without entering any air bubbles. After that, observe it under the microscope. Sometimes in the exam you will be asked to write these in 3 steps. So according to the number of steps asked, you have to summarize this. So these are the labeled parts of an onion peel. This is how you can see it is. We can say it is something like a brick wall. So these are the cells, and the cell wall is visible. Cell wall is the outermost layer of plant cells. So the cell wall is labeled, and this dark spot is the nucleus, and you can see the cytoplasm just inner to the cell membrane. And this large area which you can see in a light color or a pale color is the vacuole. So we'll discuss about these parts in detail. So you are asked to label the parts of a typical plant cell. So here I have given an already labeled diagram, but don't be afraid. You don't need to know about the plant or animal cell up to this extent at this level. When you learn advanced level biology, you will be able to learn in detail. For the moment, you need to know about the cell wall and the plasma membrane. So you can see the cell wall is the outermost layer of a plant cell, and this is the cell wall of the adjacent cell, and just inner to the cell wall there is the plasma membrane, and you can see this large structure which we call as the central vacuole, and outermost cover of the central vacuole is known as the tonoplast. Inside the vacuole we find the cell sap. So you can see in a plant cell there is a large central vacuole, and here this structure is the nucleus. Remember, nucleus is a double membraned organelle. That means the outer cover, which we call as the nuclear envelope, consists of 2 membranes. And inside the nucleus you can see there is an unorganized structure. These structures are known as chromatin network. Chromatin network. And inside the nucleus you can see this structure which we call as the nucleolus. In the nucleus there may be 1 or 2 nucleoli. So I will mention about these in detail. Then here you can see these oval shaped or rod shaped structures which are known as mitochondria. Mitochondria. This is also a mitochondrion, right? Then you can see these structures just around the nucleus and freely available in the cytoplasm are known as endoplasmic reticulum. So when there are these dot-like structures on the endoplasmic reticulum, they are known as rough endoplasmic reticulum. Here it has been labeled, and when these are without these dot-like structures, then we call it as the smooth endoplasmic reticulum. Actually, these dot-like structures are known as ribosomes. And this area, the whole area which contains all these organelles, is known as the cytoplasm. So I have labeled the nucleus, central vacuole, mitochondria, endoplasmic reticulum, and in a plant cell there is another important organelle known as the chloroplast. So there you can see the shape of the chloroplast, and you should be able to differentiate this mitochondrion and the chloroplast in a diagram, because you will be asked to label a given diagram. So actually the mitochondria has a more or less a rod shape, and here it has an oval shape. The chloroplast has an oval shape. And the internal structure of the chloroplast is shown here. So these are the main parts, and in addition to that, this organelle is known as the Golgi apparatus. Golgi apparatus. So these are the parts of the typical plant cell. Then we'll move on to the typical animal cell. So there again you will find this as a complex structure, but here you can see this is the nucleus and this is the nucleolus, and you can see the here it appears as chromosomes. The chromatin network get arranged as chromosomes when they are ready for cell division. And you can see the rough endoplasmic reticulum, or what we call as RER. And this is the Golgi complex. And the endoplasmic reticulum without these dot-like structures, which we call as the ribosomes, are known as the smooth endoplasmic reticulum, or SER. Then you can see the mitochondria by looking at the shape and the inner membrane with folds. And this is the cytoplasm. The outermost cover is the plasma membrane. So the nucleus, mitochondrion, Golgi apparatus, cytoplasm, and you can see that there are no central vacuoles. There may be small vacuoles inside an animal cell, but not a large central vacuole. And remember, there are membrane bound organelles. There are double membrane bound organelles in a cell. One is the nucleus, the other one is the mitochondrion, and the third one is the chloroplast, which is available only in plant cells. Then ribosomes are organelles without any membrane. Right, that is the labeling of a typical animal cell. And here you have to differentiate plant cell and animal cell. So the first thing, here you can see in a plant cell there is the cell wall, but it is absent in an animal cell. Then in the plant cell you can see large area is occupied by the central vacuole. There is a large central vacuole in a plant cell, but in animal cells a large central vacuole is absent. Sometimes there may be small vacuoles in an animal cell. As there is a large central vacuole in a plant cell, the cytoplasm is pushed to the periphery of the cell. Cytoplasm is pushed to the periphery of the cell. But in an animal cell, large content is the cytoplasm. Then the fourth difference is the presence of chloroplasts in plant cells but the absence of chloroplasts in animal cells. So you can complete the table by using these four facts. Then another important question, what are the cell organelles that can be observed even under the compound microscope, or what we call as the light microscope? So we can say we can observe cell wall, cell membrane, cytoplasm and the central vacuole as well as the nucleus under the compound microscope. In addition to that, in a plant cell, we can observe the chloroplasts as well. All the other organelles should be observed under an electron microscope. Next one, question number 11. The cell shows a division of labor. What is the meaning of this? So next we are going to discuss about the functions of different organelles. So division of labor means different organelles are performing a specific function in a cell. Therefore we call it as division of labor because different necessary functions are divided among the organelles. That is the division of labor. Then you are asked to write about the different organelles. First you are given the cell wall. As the specific features of the cell wall, we can say it is the outermost cover of a plant cell and it is a dead structure. The main component in the cell wall is cellulose. In addition to that, there is hemicellulose as well as pectin. Cellulose is there and there is hemicellulose. Then we can find pectin as well. And remember, cellulose is the main component. Cellulose is the main component in the cell wall. The main function of the cell wall is to maintain the shape of the cell and it provides support as well as protection to a plant cell. So remember, this is available only in plant cells. Then the plasma membrane. In plant cells, this is present just interior to the cell wall. But in animal cells, this is the boundary. We call this as the cell membrane as well, plasma membrane or the cell membrane. So remember, this plasma membrane is a semi-permeable membrane and it is also called as a selectively permeable membrane, and this is a living membrane. The cell wall is a dead structure, plasma membrane is a living structure, and it is semi-permeable and selectively permeable because it will allow only selected particles to enter into the cell as well as exit from the cell. So if we mention about the functions of the plasma membrane, it encloses the cell, it allows entry of molecules such as water molecules, certain ions, and they control the entry and exit of materials into the cell and out of the cell. Then the next structure is the cytoplasm. Before that, I would like to mention the cell membrane or the plasma membrane contains phospholipids, glycoproteins, which you have learned to a certain extent in Unit 1, There are phospholipids, glycoproteins, as well as certain cholesterol molecules. That is for your extra knowledge. Then the cytoplasm. The specific feature of the cytoplasm is it is a gelatinous liquid part. That means it is something like a semi-solid medium, a jelly-like medium, and it contains both organic and inorganic substances. The function of the cytoplasm is to maintain the shape of the cell, and it bears all the cell organelles, and it provides a medium for different metabolic processes happening inside the cell. Then we'll move on to the next organelle, that is the nucleus. So here you can see a diagram of a nucleus. So I told you it consists of two membranes, and we call it as the nuclear envelope or the nuclear membrane. And you can see there are pores which we call as nuclear pores, and inside the nucleus there is the nucleolus. There may be one or two nucleoli inside the nucleus, and you can see the chromatin network or chromatin bodies, and when these are ready for cell division, they will arrange as chromosomes. So the nucleus is the main organelle in the cell because it is responsible for controlling the functions happening inside the cell. The functions are storage of genetic material, transfer of inherited characters from generation to generation. And in the last unit, we will learn about some more factors regarding this genetic material, how they are transferred and how they are responsible for the process of inheritance. Then the mitochondrion. This is known as the powerhouse of a cell because it is responsible for, I am mentioning the function of the mitochondrion. It is responsible for producing energy by means of cellular respiration. The energy produced within the cell is produced by this mitochondrion, and that energy is used for metabolic activities of the cell. Actually, the metabolic activities are all the chemical reactions involved in maintaining the living state of a cell or a living organism. These metabolic activities are of two types, known as catabolism and anabolism. Catabolism is breaking down of molecules to get energy, and anabolism is the synthesis of molecules. So if we talk about the specific features of the mitochondrion, it is a double membrane organelle. It has a rod shape, and the inner membrane has folds which we call as cristae, and the inner part of the mitochondrion is known as the matrix. You don't need to learn up to this extent. Just remember, and these folds will help you to identify the mitochondrion properly. Then the Golgi complex. Golgi complex is again a membrane bound organelle. It is just like sacks stacked on one on another, and this is associated with a secretory function, and there are secretory vesicles surrounding the Golgi apparatus or the Golgi complex, So the main function of this Golgi complex is to produce secretory substances. They will produce secretory substances, store these secretions, and they will transport those secretions. So these secretions may be enzymes and other proteins. Then we'll move on to the ribosome. Remember, ribosome is an organelle without a membrane. It consists of a large subunit as well as a small subunit. So these ribosomes may be available freely in the cytoplasm, or they are attached to the endoplasmic reticulum. So in here you can see the ribosomes attached to the endoplasmic reticulum. The function of the ribosome is to synthesize proteins. Then we are moving on to endoplasmic reticulum. So it is again a membrane bound organelle. It is a network made up of flat or tubular sacs like this. And when ribosomes are attached to this endoplasmic reticulum, we call it as rough endoplasmic reticulum, or RER. The function of RER is to transport proteins which are produced by ribosomes. When there are no ribosomes attached, we call it as the smooth endoplasmic reticulum. The function of SER is to synthesize lipids, steroids, then transport these lipids and steroids within the cell. Then we are moving on to the vacuole. The specific feature of the vacuole is it is a large organelle filled with a fluid. You learned that the outermost cover of this vacuole is the tonoplast, and the liquid filled is known as the cell sap. So this cell sap contains water, sugar, ions and different pigments. You learned that there is a large central vacuole in a plant cell. In animal cells there may be small vacuoles. In unicellular organisms we can observe contractile vacuoles. The function of the vacuole is to maintain water balance, support and provide color to the cell by the pigments available. As the last organelle, we are going to discuss about the chloroplast. So chloroplast is again available only in plant cells, and it is responsible for carrying out photosynthesis. Since grade 6 you have learned about this, the function of photosynthesis. So the chloroplast has an oval shape like this, and it is a double membraned organelle, and the basic parts which will help you to identify the chloroplast is those grana, these stacks of coin-like structures, grana. So you will learn further about this chloroplast and the function of photosynthesis in advance in your advanced level. Then we have to discuss about cell growth and cell division, as it is another lengthy part. I think I will have to discuss it in another presentation. So hope you got a clear idea about structure and function of different organelles in a cell. So you can subscribe my channel